Now I'm assistant professor of nematology at Clemson University. I started my position in January, and after 2 months, we were hard hit by pandemic. So I pretty much had to stay in the home and do my all research from computer for 2020. In 2021, I was able to get out into the field and do some research. So I have very few data, very preliminary data, preliminary analyzed. So I'm going to talk about that a bit. And my topic would be nematodes associated with peach, mostly in southeastern US, and some of their management challenges. So plants are attacked by several um, types of pathogens. They could be nematodes, they could be fungi, could be bacteria or viruses. Nematodes are probably the biggest plant pathogens. So they were identified very early, around 1743. That was the first time when plant-specific nematode was identified. And we started having sophisticated compound microscopes and electron microscopes, and then we discovered virus, viroids, those small particles. And just to show you the size of each pathogens, uh, this is the this is the head of the nematode, uh, and you can imagine how big these nematodes can be. So if a nematode goes inside the plant tissue and finds a good cell, it can pretty much invade that entire cell very quickly. If you pick a handful of soil from any agricultural land or any land that has some plants, you will end up having a lot of nematodes. And when I say a lot of nematodes, that's hundreds of thousands of nematodes in a handful of soil. But all nematodes present in that soil are not harmful to our plants. There are some nematodes, like free living nematodes, that constitute 25% of the entire nematode population. They are very good for our soil health. They maintain soil health by feeding on antagonistic fungi and bacteria and help release nutrients into the soil. So these free living nematodes are very important from uh, soil management and crop management perspective. A lot of nematologists, they just focus on plant parasitic nematode, which constitutes just 10% of the entire pie. They are really important because they cause huge economic losses, but we should also be looking for free living nematodes that really help maintain our soil health. For my data, I'll be presenting some data on free living nematodes and plant parasitic nematodes. When I say plant parasitic nematodes, those are the ones that have the stylet, a distinct stylet in their mouth part. You can see it here. And some have knobs, some do not have knobs. But we look for this stylet to identify whether the nematode is plant parasitic or free living. And a lot of times we don't realize that nematodes are a big problem, but when a susceptible crop is present in a virulent nematode type, there are several genus are present, they can cause a huge economic losses. And they are just not just another can of worms. They are really important. And we have a broad audience, so I just wanted to give a brief perspective on nematode. Uh, just to be very brief, nematodes are highly diverse in their size and shapes. They are microscopic organisms, so they are still microorganisms, but they are bigger than viruses and bacteria and even fungi. We cannot see them without help of microscope. And some of them are ectoparasites. That means they feed on plant roots from outside externally. and some go inside the host tissue and start invading the host. Nematodes, those plant parasitic nematode, they cause hundreds of billions of dollars losses every year worldwide. And for peaches, there are few nematodes that are very, very important. Um, in general, nematodes can attack pretty much every species of crop on Earth. There are no any crops that are not attacked by one or other types of nematodes. So if you have any agricultural land or any crop, just imagine you will have some sort of plant parasitic nematode. It depends whether that nematode is highly damaging or not, but you will end up finding those nematodes in there. And on peaches, I'm gonna go to these some of the common symptoms in next slide. But uh, since this is a perennial crop, it is sometimes very difficult to see the nematode damage. And for that, we typically look for the uh, dieback symptoms like the twigs dying. And when we see those, we go ahead and uh, sample the soil and look for the presence of nematodes. Some of the common symptoms of nematode attack, like above ground symptoms, could be chlorosis, yellowing of the plant leaves or foliages. Plants get stunted and lose their vigor when the nematode is present in very high number. They can cause wilting. That's typically true for root knot nematodes. And dieback symptoms, like the picture I showed in previous slide. The below ground symptoms could be swollen roots, could be galls or knots if they are root knot nematodes. And sometimes we see necrosis and lesions, primarily in the storage roots. And a lot of time we do not realize that nematodes are damaging to our crops because a lot of these symptoms, either above ground or below ground, they are very similar to um, other symptoms caused by other pathogens or even some abiotic factors like nutrient deficiencies, water stress, all those things. So in most cases, losses to nematodes are attributed to other pathogens. That is why we are seeing like very low figure for total nematode losses to our crops. And that is the reason why nematode damage usually goes unnoticed. The picture is a peach tree short life. The one in the front was attacked by one of the plant parasitic nematode called ring nematode. When that nematode is present in very high number, that can cause stunting of the plant. That just stresses the plant, and that makes vulnerable to bacterial infection that eventually leads to peach tree short life. And when these trees get that peach tree short life, trees can die in 3 to 4 years. So growers plant their peaches, wait for production in third or fourth years. And if the nematodes are very high and they get peach tree short life, they can like lose their entire crop. So that's a total loss. For peaches, there are three nematodes that are very, very important: ring nematode, uh, dagger nematode, and root knot nematodes. And their action threshold is very low. For ring nematode, if we see 20 nematodes per 100 gram of soil, that triggers the alarm for management. Um, the number goes like 50 for dagger and root knot 50 per 100 cubic centimeter of soil. And this action threshold can differ from type of the soil too. This one is for sandy soil, and if the soil is clay or loamy, the number can be little higher. But still, they have very low damage threshold. So that is the reason why we encourage our growers to submit their samples so they can start management action earlier. And for root knot nematode, there are several species of root knot nematodes. A lot of our peaches are guardian rootstock. So that guardian is resistant to a lot of root knot nematodes, except for the new one, which is called peach root knot nematode, Maloidogyne florensis. So guardian lacks resistance to Maloidogyne florensis, but it is resistant to other species of root knot nematode. And guardian is also tolerant to ring nematode, but it is not resistant to ring nematode. So even if you have a guardian in your orchard and have a very high number of ring nematode, the tree can very likely to sustain a damage. This is the ring nematode, very easy to identify under microscope. Um, a root knot nematode juvenile and dagger nematode, that's a relatively bigger nematode. So when we find some peat samples or I do my research, I first look for these three nematodes and then go for other parasitic and free living nematodes. So what are the common management options we have for these nematodes on peaches? There are some chemical nematicides, fumigants or non-fumigants. Uh, we are shifting towards non-fumigant nematicides because of the adverse effect of those fumigant nematicides on human health and environment. We have some biological nematicides as well, and some cultural method that is probably the best method. I'm going to talk about that why in my later slides. And there are some legislative actions. I'm not sure how much we can apply this option for managing nematodes and peaches. That's going to be really tough. There are some host plant resistance options, but like I said previously, uh, the most widely planted guardian rootstock is resistant to most of the nematodes except for peach root knot nematode, Maloidogyne florensis, and that is just tolerant to ring nematode, not resistant to it. Some of the cultural methods that we can use are use of soil amendments. Uh, Peach is a perennial crop, so crop rotation is not really a good option. Um, some of the other methods would be use of some cover crops. Um, I'm conducting some research on cover crop, but I'm not covering that in this presentation. I do not have complete data for that. Hopefully, I will have that data to present next year. And cleanliness of equipment, farm equipment is really important to avoid contamination of nematodes from one field to another. Um, I had initiated a nematicide trial in one of the farms in Regis. Um, and the variety was Scarlet Prince. So I used like 6 different combinations of nematicides, Videt, Vellum, and Magistine. The full rates, first three were full rates of uh, nematicides, Videt, Vellum, and Magistine. The Magistine is a biological, Videt and Vellum are chemical ones. And other treatments, 4, 5, and 6, were combinations of half of the recommended rate of those 3 nematicides. And I had one uninoculated, untreated controls that did not receive any nematicides. And that was randomized block design with 5 replications and repeated in 1 month interval. Um, the nematicides were drains applied with watering can in 1 square meter on the berms of the field. That was a very small area, but that was enough for nematodes because nematodes, they don't move very fast in the soil. Once they find the root, they pretty much stay in there and invade the plants. And nematode samples were poured before nematicide applications and month 1, month 2, and month 3 after nematicide application from that treated area in the around the trunk of the peach tree. And these trees were around 3 or 4 years old. so relatively young trees. There was some interactions in first and second trials, first and second experiments for free living nematodes. So I analyzed them separately, but there was no any significant difference among these different combination of nematicides on abundance of free living nematodes. Um, Month one is the sampling before nematicide applications, and month two, month three, and month four were like each month after nematicide applications. So month two, we observed relatively low number of nematodes in each treated uh, ones or for each treatment, and the number spiked up for month three and month four. But there was no any significant differences among those treatments. That can be good, like when we talk about free living nematodes. For the second experiment, there was not significant like the effect of those treatments was not significantly different for all the sampling times except for the third month. Um, um, Videt and the mixture of Videt and Magestine did not significantly reduce the number of free-living nematodes compared to the uninoculated control. All other combinations or sole applications of nematicides, they significantly reduce the free-living nematode community. That's just the third month. On ring nematode, like we observed some lower number of ring nematode on the month two, that is 1 month after nematicide application, and the number kind of started spiking up again later in the months, but there were no any significant differences among those treatments for each month of sampling. On total plant parasitic nematodes, the result was the same as for the ring nematode. We did not see any significant effect of those nematicides on abundance of those ring nematodes. So these nematicides, I'm not saying that they are not effective, but I'm trying to say that in my experiment, they did not seem to be very effective in reducing the population of those plant parasitic nematodes. Maybe we need some better chemistries to lower the populations of plant parasitic nematode on peaches. Um, I was thinking like that 1 square meter area was kind of a very small region to study. So we decided to do an experiment with larger area in our farm, the Musher farm. So I picked like one nematicide, velum, and applied that at two different rates. Uh, the recommended rate 1x and twice the recommended rate 2x, and that was applied with a sprayer, and that was applied around the canopy of the tree, that large area which spans 25 to 22 feet. And data were collected before nematicide application and 1 and 2 months after nematicide application. Again, both rates of velum 1x and 2x did not have significant effect on free living nematodes when compared to the control. But again, in month two, which is 1 month after nematicide application, the total number kind of went down and then started going up in subsequent months. Like month three, which is 2 months after nematicide application, we observed like it's going slightly up. Um, for ring nematode, there was no any significant difference even for those two different rates of velum. Um, but the numerical number was lower for month two, which is 1 month after nematicide application, and month three when compared to the um, control that was no nematicide. On dagger nematode, uh, there was again no significant differences among the treatments. They did not really suppress nematode populations. Rather, we saw increase in the populations of dagger nematode in subsequent months of nematicide. Total plant parasitic nematode, same trend as a dagger. We observed increase in abundance of these plant parasitic nematodes rather than going down after nematicide application. So whether the area was 1 square meter or like 10 square meter, uh, we I pretty much got the same kind of result. We did not see any significant effect of those nematicides on free living as well as plant parasitic nematodes. Um, we also started some soil amendment trial. Before I joined Clemson, Guido had started a soil amendment trial at the mushroom farm of Clemson University. And that was started, I think, in 2020. But I missed that year and I jumped to the next year in 2021 and collected. So that field that has like 9 different rows, and in each row there are 15 trees, 5 of each tree. So that's 9 rows and 3 replications, and basically one tree is like uh, five of the same trees in the same row. I'm not sure if I explained that very well. And spacing was 16 feet by 22 feet. There were 3 varieties of peaches: Big Red, July Prince, and Ruby Prince. And we used 3 mulch rates. The first rate was, we say 1x, which is 8 feet width. The depth of the width of the mulch was 8 feet, and depth was 6 inches. For 2x rate, same width, but we increased the depth to 12 inch or 1 feet. And the gold standard was like without receiving any mulch applications. And the data was somehow interesting. The If we look at the like before and after mulch application, there was some differences among those numbers of free living nematodes. So the blue is 1x mulch treatment, and the orange bar is 2x mulch treatment, and the gray bar is a control which did not receive any mulch application. And these 1x and 2x rates had significantly higher number of those free living nematodes. So that field had received those mulch from in 2020. So the initial number of free living nematode was already high in the plot. After treatment, the number seemed to go down, the overall total number, but still there was some differences. Um, 1x and 2x were not significantly different than the control, but there was significant differences among 1x rate and 2x rate. And 1x rate, 2x rate seemed to like have higher number of free living nematodes, significantly higher number of free living nematodes than 1x. On plant parasitic nematodes, that was very interesting. So before treatment, the number of these nematodes uh, among those treatments were not significantly different, but after treatment, that was significantly different, but not as expected. The control had, well, I would not say not expected. The control had a higher number of dagger nematode in relation to 1x and 2x treatment. So that actually worked well in suppressing dagger nematode. In total plant parasitic nematode, kind of same trend. 1x and 2x seem to suppress total plant parasitic nematodes, compared to the one that did not receive any mulch. So the take-home message from this mulch trial is that mulch, they may not suppress, they may not increase the number of free-living nematodes, which are good nematodes for soil health, um, but they seem to suppress the plant parasitic nematodes. So it is probably a good idea to apply mulch in the orchard, peach orchard. The overall message from my talk, ring, dagger, and root knot nematodes are the most important nematode types for the southeastern US. There has been a lot of research going on for the management of these nematodes on peaches. We thought Guardian is a one in all solution for all nematodes, but turned out it is um, not very good for some of the root knot nematodes like peach root knot nematode and ring nematode. When the number is very high, you know, the peach can sustain a high damage. Um, Use of chemical nematicide is still a preferred method, but unfortunately, I did not get very good result, and that follows the same trend for probably most of the other field research. So there needs to be done some work on new chemistries or better chemistries on those nematicides for managing plant parasitic nematodes. Um, and probably the soil amendments for now would be the best options. It would be very best to have some resistant cultivars resistant to all nematodes, but that's gonna take a while, a few years, I would say. But before then, probably use of the soil amendments can help suppress at least those plant parasitic nematodes. With that, I would like to thank you all for your patience. And if you have any questions, I would be happy to answer. Great. They can be applied like a foliar application or soil application. It could be. No, we did not spray on foliage. We sprayed on the ground. For the second trial that I talked, that was the drench application, but with a sprayer. We wanted to cover the larger area, and when we had a sprayer, so we used that. The the first trial was the sandy soil, but the second one was loamy soil, clay loam. Yeah, and that can affect the result. Yeah, I agree. Yeah, that was the regular mulch. What was that, Guido? That's a good question. We have been using those thresholds from a publication or a guideline that was prepared in 2000. So that was 20 years old, but that is still is valid. So we rely on that guide. So that has information for all crops, mostly all, like most of the crops and most of the nematodes. And we use those numbers to see like how damaging those nematodes could be. I agree. Yeah. 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 Well, to me, like for all crops, it's just one. But no, We rely on that guide. Probably we are using the same guide. That's the minimum number. That's for other root knot. When we talk about peach root knot nematode and guava root knot nematode, which is an emerging nematode, the damage threshold is really low. I would agree on that one. Yeah. Okay. All right. Thank you so much. All right. There you go. Thank you. She's gonna share.
Okay, next we have Dr. Brent Blau, University of Georgia, giving us an update on insect and management and teaching. Thank you, Dr. Blau. Awesome. Thank you, Andy. And thanks for all of you attending today. It's great to see you all in person. I know things are still crazy out there, but thank you for making your way here. Um, so I'm gonna really just start off kind of giving off just a couple things to look forward to in 2022. Not necessarily good things, but at least things to keep on your radar. And then I'll talk about a little bit about more of the updates from the season, looking at San Jose and then Orioles fruit loss. Right. So things to look forward to. Positive things. Um, we're gonna a little delay there. Sorry about that. Positive things are some of the projects that we're going to be working on from in my lab. Um, Part of it is because we really need to the borer management, looking at new chemistries and new ways of getting those out there to look at peach tree borer and lesser peach tree borer. Thrips management, I know that's a big issue for a few of our growers. We're working with the mustard farm to actually bring in thrips so we can do a really nice spray trial for thrips. Because that's something that we've been struggling with for a while on how to actually do this. We're going to really push through to some data on how to manage thrips. And then scale. Scale is always the elephant in the room, trying to figure out what we can do with this, looking at some new adjuvants and some heavier oils to see if we can get really smother these guys to kill them. Now, the bad thing to look forward to in 2022, depending on who you are, is the pure false ban. Right, so starting March 1st of this year, all tolerances of chlorpyrifos for fruit uses will be revoked. That means if you have any chlorpyrifos on hand after March 1st, you will not be allowed to use it. All right, so this whole thing happened about was now 4 months ago. Kind of came as a surprise. We all knew that the ban was coming, but how fast it came was kind of a kick in the shins. But note that after March 1st, for beer costs. So that's going to mean there's going to be some changes in a lot of our management recommendations for Petri board and lesser Petri board. We don't have anything that's going to be necessarily a one-for-one change or like a replacement yet. But this is definitely something we're going to be looking at this year. Some of the recommendations that we have that still work relatively well, Asana or Exavalorate and Ulticore are going to probably be the two closest of actually just or as a replacement spray. Ryman works relatively well as well, but this is an insect growth regulator, so it's a little bit slower to act. You won't necessarily see that direct knockdown, but it still works well. Has long. We also have mating disruption. So mating disruption works really well. The caveat to that is it has to be on a large acreage. Basically all your peaches that are in that acreage have to be under disruption. So if you have one block next to another block, the one block is under mating disruption and the other one's not. Mating disruption is useless in that situation. You have to have all of those peaches under disruption for this to actually work. Also, can take a few years to see some of the benefits from this. So it's long. It's a long-term management program, but it can work well for situations. Then we have the entomopathogenic nematodes. Romani was talking about some of our bad nematodes. These are the good nematodes. They will attack our insects. They do not attack the plants. And data from the USDA has shown that these work really well, especially for peach borer, because you can apply them to the soil. The nematodes then get into the soil and attack the peach borer in the roots there. It doesn't work as well for electric peach borer because it's on the actual trunk or on the scaffold limb there. They can fly out, but they're working um, for at least for peach borer, really well. And that's awesome about that is that's a curative as well as a preventative. Scale. Scale, we still have some really good insecticides that work well for San Jose scale, like Centaur, Esteem, and Movento. We still can use our horticultural oils, using some of our softer things like Venerate and Sea Light as a contribution. All have nice impact on San Jose scale. So boozing for pure phos was one of the methods we did have for San Jose scale, but we still have some other things that work really well. But that's still that the other options up with the question mark is saying we still haven't figured this out completely. So we're going to definitely our San Jose scale management options. And then the 2022 guide has been updated to reflect the changes for PureFOS. Those guides have been printed and will be available soon. And I'm gonna, if hopefully everyone here is signed up to receive messages from the blog. And if you're not, the UGA Peach blog, we update that not frequently, but really we update it to reflect really important information. And so I'm going to update that soon to give you the link to the new 2022 guide so that everyone will have that in time for this new season. All right, so let's move on to San Jose scale. So one thing that I've been working on for the past couple of years is mating disruption for San Jose scale. I know some of you have tried mating disruption for peach borer and oriental fruit moth, and it works really well for those moths. Well, there's a sex pheromone for San Jose scale as well, and so I've been really interested in seeing, can we use a pheromone to disrupt the mating for San Jose scale and hopefully also reduce problems from scale. So working with a couple of companies, CBC America and Trace A, we came up with this design quarter-acre blocks of peaches, 3 drops for each of our treatments. Tested 3 different formulations and mounted both the adult males. The adult males of the San Jose scale are the flyers. They can actually move around. They're the ones we're trying to disrupt. We're trying to stop those males from finding the females so that mating doesn't happen. All right, but then we also look at the crawlers. The crawlers mean those are the babies. So if crawlers exist, that means mating took place, which means that we didn't necessarily completely disrupt the mating. All right, so here is our treatment. We had our untreated control, which we were still treating for other insects, but we didn't use any scale focused insecticides. Then we tested isomate, which is one of the common formulation that's used for mating peach before. Then we also experimental from Trace and applied them prior to bloom, at least in these settings. All right, just an example of what what we can see when sampling for males. If you've never sampled for males, it's pretty crazy. This is under mating instruction, and all those little dots, this is all massive, are the same. This was alarming to see is the difference that we're actually seeing. If we're looking at this throughout the season, early in the season, we're seeing really no males in any of our treatments. We get our first capture in our control early April. For our estimate, we actually almost a month later, late May, is when we first got our first capture. That tree stage 2042 was early May, and then late April for the tree stage 2043. That doesn't really mean much. What it does mean is that we're not getting complete trap shutdown. We are getting those males on those traps. And that trap that I showed you, we are catching quite a few. But yeah, as you can see throughout the season, we're seeing fewer for all these treatments compared to control lines. We're getting almost 2,000 of those males on the card. And actually, what this doesn't show is times that by 5 to get the actual numbers. That's huge. Right? So here, looking at our average scale per card throughout the whole season, we're getting over 1,000 per card for the control. All the other treatments, both the three different mating disruption formulations that we tested, all had significantly fewer males. So that is really encouraging. We're not getting the complete trap shutdown, so mating does take place, but we're reducing that by a lot, which it gives me hope for the future. Right? But the big concern is looking at these crawlers. So we know that the males are flying, we can catch them. Is that mating taking place? We use double-sided sticky tape wrapped around branches. All these little dots here are the crawlers. What we get, it looks something like this. This is pretty good. All that yellow though are crawlers. All these little circles are those crawlers. It's alarming as well. But how does it look? So caveat to this is I only have the first part of the season. I only have through May of the data here, but through May it's looking pretty good. We still have in our main instruction treatment. It's not gone, but it is a significant reduction. We're going from an average of 350 crawlers per 1 down to about 100. So that's still quite a few, but it's definitely a lot better than so I can't yet say that mating disruption is the answer, method that hopefully better develop how to use it, when to implement it, and maybe actually get these much lower. Or similar to other mating disruption where it's going to take a few years of use before we actually can get these bars down to zero. All right, so mating disruption is not our answer yet. So we also look at within season use of light rate of horticulture oil. So this was done at a single orchard, single variety. We used a 0.2% dam oil added to our standard spray program. So every time we being the grower, I didn't do any of the spraying. I just monitored the scale. The dam oil was added to that spray. Did it 5 applications and monitored the San Jose scale crawlers. Note here that there were some issues with mixing with the sulfur, mixing with that oil. So I'm not saying don't spray your fungicide, but you might have to spray sulfur separately, or work on a way to pre-mix that so that it can actually spray. We didn't see any issues with phytotoxicity, but it's more of a mixing issue. All right, so here's our field. We had our control, no oil, but still had that standard management program, and that 2.2% oil here in the blue. We monitored scale on 5 trees at 4 different sites on both of those. And here's what we saw. There's no statistical significance between these two treatments, but we did see a numerical reduction in the number of scale in that oil. All right, so statistics is one thing, but in real life, seeing that reduction is really encouraging. If we can get that reduction year after year after year, similar to what I was mentioning with the mating disruption, we might be able to really knock that scale down to a population where we're not seeing any damage to the trees or to the fruit. And also having no issues with the phytotoxicity, at least last year, with those 5 applications, the fruit looked good, the leaves looked good. I'm really encouraged by So in summary, I didn't mention that, but dormant oil applications are still a critical part of our management program. If you have problems with scale, doing those two dormant and then delayed dormant applications of oil is very, very important to keep our scale populations under control. Even with those, we're still seeing issues popping up. And so make sure that your coverage is good. The timing, making sure that you're adding oil right time. So if you're doing a pre-pruned application of your delayed dormant, pre-pruning, you want to increase your gallons per acre. If you can range up to about 200 gallons per acre, I know it sounds crazy, but you're going to get better coverage that way. The better coverage equals better control. Now, if you're adding the oils to trees that have already been pruned, you can reduce that rate down to about 100 gallons per acre and still get really good coverage. So, trying to think of when the timing of your applications could also impact how much you're applying. Adding in the insecticides, especially to that delayed dormant application, can improve management. So Esteem, Centaur, Venerate all can help reduce or increase the efficacy of that oil application. Mating disruption. So I can't recommend it yet. Well, it's not. You can't buy it yet. It's still under experimental stage, but it's exciting. It's had some really exciting results that I think within a few years, hopefully, we can have a better understanding of what this can do for our industry. And then similarly with this within season spray of the low rate of oils, can't recommend that yet either, but it's still something to look forward to. It's exciting. We have really good data showing that we can reduce that scale without really harming the trees, but it might take several years to actually see the true impact from that. Also, we really need to look at how this impacts how these oils can work with Captan. Guido's done some work with that. I did some work last year with it as well. It's not necessarily going to be, yes, you can use it with a Captan. You need to figure out what rates will work together. So, again, this works really well. I can't necessarily recommend it. It's not in the Peach Guide yet, but it's something to look for. All right, and then our last test to talk about today is Oriental fruit moth. Generally, Oriental fruit moth is more of an issue in the upstate. I think part of that is just the climate is a little bit better for these moths. But where it's a problem, it can be a huge problem. The moths emerge and mate shortly before bloom, and then are basically out there for the rest of the season. Different generations throughout that season. You can have up to I think 6 generations in the season. So they're out there and they are active. What's really nice is that early season insecticides that we are applying for plum curculio normally will provide excellent control for Oriental fruit moth. But what we're seeing is late in the season, though, unfortunately, cultivars that are ripening after June, which is not even that late, we're starting to see the larvae get in that fruit. That's the big problem there. We don't have necessarily, or we won't have any evidence of insecticide resistance yet, but we are seeing where it appears to be that some of the things, like the pyrethroids, are not really getting the control that they used to get. And so we really need to focus on based on growing degree days. And timing seems to be the most important part for Oriental fruit moth, and that's in our management guide. I'll show you in just a second. But if you are in the upstate and you have had problems, mating disruption for OFM might be one of your best bets. All right, mating disruption. There's several different companies that produce it. Two that I'm familiar with are Isomate OFM T and Cytrex, by CBC America and Cytrex by Trace. Both of these have really good results in the field. Both of them are going to work a little bit better on a larger acreage. If you just have 100 trees in your backyard, a mating disruption is probably not for you. But if you have over a 5-acre blocks that are not all surrounded by trees or surrounded by other pome fruits and stone fruits, then mating disruption could be for you. Because like I mentioned before, with the peach tree borer, mating disruption, if you have other trees, other stone fruits, other pome fruits around, all that has to be under disruption. Because if there is an area where these moths can mate, that mated female can fly wherever she wants. She doesn't have to stay in one tree that's not under mating instructions. She can be over in your neighbor's field mating and then come over to yours that is under mating instructions. So you need to have everything within your area under mating instruction for it to work best. And if it's something that's new to you, you haven't used it in the past, you might need to have a transition year where you use mating disruption as well as chemical management to really knock down that population so that the following year mating disruption can work all on its own. And just because things fall apart, things don't always work 100%. It's still advised to monitor if you're using mating disruption. Monitor for flagging damage, which is this one. See those tie back and get a little curving, look for little dead flags on those trees, or look for the damage on the fruit. You see the frags where the larvae have crawled in and dumbing on that fruit as well. With looking at about 20 shoots per 20 trees, if you get about 1% of fragging when using mating instruction, then you have a problem and work using an insecticide to treat those. If you find any infested fruit when you're using mating disruption, you're going to want to spray. All right, so mating disruption is not going to be for everyone. It takes a lot of effort to put them out. Right? So it might be for you or whoever. Thankfully, if we're using one everyday models that are in our management guide, using the chemistry we have. So using best to figure out what we're doing with these OFMs is we have one pheromone trap for about 10 acres. See if you get 10 moths per trap after that third cover. Using a everyday model during time that's going to be the best. Using that first trap, if you more than 10, we'll complete our biome. Then we know we need to start calculating growing degree days. We do have several good insecticides that work. So this is what we're going to see in our guide. These are the degree days that we need to keep attention to. That biofilm that's there. So when you drop that trap and you catch that first sustained trapping of moths, that initiates biofilm, and then we start to calculate our growing degree days. The guide will help you with that. And then these are going to be our timings for our management. And if you can follow that, which I know it's easier said than done, but following that method can work really well. The insecticides that are in our guide are here. All of them are quite good, especially those pyrethroids. But like I mentioned before, the pyrethroids in some cases not be working as well as they had in the past. There's no real evidence for insecticide resistance. I'm not sure what's going on. But because of that, it's always a good idea to rotate. We have some other chemicals. We have some diamides, spinosins, organophosphates. We can rotate that work really well, but I wanted to look at some other potential options, see if we can really increase our chances to limit any potential for insecticide resistance. So I added a whole new chemistry, spear left. I won't even get into it. That will kill the moths. When using that as rotational with one of our good pyrethroids, it's mustang max. Also looked at another diamide here, Alcor, doing that as rotational with mustang max. And Emidan using our standard spray or standard rate, but then also can we reduce that? I know Emidan works well when it does work, but it's also a little bit expensive. And so if we could reduce that rate, maybe we can management or efficacy from that. And one key thing I need to point out is that Emidan has to have a good pH, something lower than 6.5. If you have a really basic water, Emidan is basically stop working. If you've had issues in the past of Emidan not working, make sure to check the pH of your water. That's an easy thing to do. It's something I always forget to do, but when I'm spraying Emidan, making sure to check my pH. And also look at other diamides, see if you can throw those in there. fruit farm. Unfortunately, there's not a huge pressure from OFM. Unfortunately for me, for doing this data, there's not a lot of pressure from OFM. We were able to get some good results. Looking at the flagging, we found that pretty much everything that we tested reduced the percentage of flagging on those trees. Imidane at the low level was not from our control, which is here in the yellow, but it still numerically reduced of damage to those shoots from OFM. That's encouraging that even at that low rate. But everything else we did significantly lowered the amount of flagging. Now, I totally skipped over the reason that we're concerned with the flagging. That flagging is actually produced because the moths are laying their eggs in that shoot tip. Right, so then those eggs will hatch and the larvae will develop in that shoot tip. So even though that's not damage to the fruit, those flaggings really mean there's going to be new offspring that could potentially attack the fruit later in the season. All right, but we also did look at fruit infestation. This is what I was expecting to see. But when we're cutting into the fruit, I was unable to find the larvae. Probably part of the timing of the larvae had already left, but we still would find these cracks, this really gross stuff inside the fruit. So I looked at least, I didn't find any larvae, so there was no 100% evidence of OFM in these fruits. But I did count this damage. We called that fruit infestation. But what we saw here is that in the untreated controls, we're getting about 16% of the fruit had this damage on the inside, whereas our Alcor, the diamond Mustang Max, that spear left of Mustang Max, as well as Emiden Standard, had no damage on the inside. All completely clean. Same with X-Row. The vertebrae, the no diamond at the low rate, we were still seeing that damage on the inside. In previous years, I tested vertebrae at a higher rate. Higher rate was good, but this low rate is not. Low rate for OFM, not worth it. Emidane at the low rate, we still saw a little bit of damage, very low, what we call 1% there. 1% is more than we want to see in our fruit. So Emidane low, right now it's first year, I'm not sure what to say about that. But it's Emidane on its own, it's standard, still works really well. All right, so our summary here, main instruction isn't an extra option. If you've, especially in the upstate, this works really well if you have an isolated orchard, not surrounded by any other palm or stone fruits. Main instruction can work really well. But for either using main instruction or spraying alone, monitoring is important. Monitoring is what really triggers a lot of our management timings. I know it's not fun to go out. That's what I do for a living. I know it's not fun. But it can really help time those applications if you have problems with OFM. And while we haven't seen any insecticide resistance yet with OFM, rotation is still important. It's just a good practice to get into to make sure that we don't have problems later on. So the Spear Left, which is a new product for us, seems like it'd be a nice rotational product that we can mix in, or not mix in, but using our rotation to help reduce any resistance there. Diamides seem to work really well when used in the standard rates. Same with Imidane. At standard rates, we got really good control. Again, timing is crucial. The 3-day model is in our management guide. It's in the 2022 versus all not just the 2019. But if you're looking at that and you never have questions, please let me know. Science is what I do, and some of this math is me as well. So if you have problems with it, please let me know, and I can try to help you guide through that. All right, and then just that we have for this year, spray guides like I mentioned, expect to see those soon. It is online now. I'll be posting about that on our blog, the UGA blog. If you haven't gone there, go there. I highly recommend it. We're not going to spam you. You're not going to get all these alerts that UGA Peach Blog. I don't know. You go on into it. It's not like social media. You get important information at important times. And then of course the Mario Game smartphone app, which is my bloodline. I love it. If I need any information, I usually can find it there. It's great. All right. Well, thanks my lab for getting all this data together. My grower croppers for me to do all this stuff on their land, and of course all the funding crucial to get anything done. So thank you everyone. Thank you all for listening. We have not looked at copper and oil. Um, that's a good question. Not have not. So I didn't mention that this was it was we had a four we were targeting those, but I did not target like we did not do a specific target. I was trying to get a whole range. Sorry, I did not cover that in there. We had four different applications targeting different topics. Right, and I have not seen anything looking at the adjuvants. So right now, our three top chemistries that haven't studied is our um, Sana XL, Alt4, and Ryman are our top three right now. I have not looked at it, and I have not seen anyone looking at the adjuvants. That's a really good question. That would be great if we actually extends that residual. That's a very good point. The cost is going to be high for a lot of these alternatives. Mating instruction is expensive. The Tryman is also expensive. Asana is a little bit cheaper as a pyrethroid, but everything is going to be more expensive than for purifiers. That was one really nice thing for purifiers. For what it was, everything we have is going to be more expensive. The nematodes are actually relatively inexpensive, especially if you're using it for pastry board. The cost analysis for that is their cost benefit is pretty high. It's low price point compared to some of our last question here, if we could. Good to know. Spider mites, that's the oil is great for that. I didn't realize the steam pumps. That's cool. Awesome. That's even better. Well, if we could thank Dr. Blau for all his time and effort, I appreciate you. Thank you. Next, we have Dr. Phil Brennan. I've gotten the presentations mixed up a little bit there, but Dr. Phil Brennan is with the University of Georgia, well-respected plant pathologist. Appreciate his time and effort. Looking forward to hearing from you. Dr. Brennan.
Yeah, Guido and I work together enough. We could probably give each other's presentation, but it might not be quite right. So I think it's good. I got the right one up here. All right, so I'm going to be talking to you about some of the disease management stuff, kind of updates on some of the latest stuff that we've done in the last year, and bring that to your attention. We have developed a pretty good spray program here for the southeast, and it works pretty well as far as fungal organisms. Guido actually initiated putting a scientific paper together. He was gracious enough to let me be on it. But this kind of gives the details of what we do with this spray program, and we do think it actually has worked pretty well for y'all. But basically, the critical thing you've got to get out of these spray programs is we're mixing a lot of different frac groups. If you look at that, there's a lot of frac groups involved in that. We try to utilize the old chemistries as much as we possibly can, the ones that don't develop resistance like Bravo and Captan and sulfur. they're thrown in there, and that's critical to the program. So when I'm going forward for the next few minutes, I want you to keep in mind that that's one of the reasons we really don't want to monkey with what works. If it works, we don't want to break it, and I'll make comments on that. But this has worked pretty well overall, but we still have some challenges. We'll talk to you about brown rot at first, and just some of the stuff that's come out in the last year or two for brown rot. Just to review what we do, I don't think I talk to you all a lot about this every time we get together, but for our research trials, we're trying to do everything in a bad way for a lot of reasons. We want a lot of disease. So we do some things that are pretty weird from the standpoint of what y'all would do. But at the same time, in our trials with the USDA and also the ones we do in Athens, we actually use air blast sprayers. We're putting out about 80 to 100 gallons of water, similar to what you guys would do, and using an air blast sprayer. We're also putting out, whether it's scab trials or whatever, similar type rates. And then for the brown rot trials, we're going in about 14 and 7 days before harvest, putting out these applications, and then we will go in, collect fruit, and let them rot over time and see what we get out of that. So that's how we do these trials. Just looking at a couple of trials from 2020, I want to concentrate on one or two things here. Hard to get at this from this angle for you guys, but if you look up here, here's the untreated control, okay? So you can see what you're getting out of that with those two applications prior to harvest. And then you can see also what Maravon does. Again, Maravon to me is our best fungicide still that we have on the market right now. It absolutely is the best. There's some other products that are comparable. When you look at Luna Sensation right below that, you can see it's very comparable in both of those trials from 2020. But the other thing I wanted to concentrate on is looking at the what we normally recommend is not back-to-back applications of Maravon such as you have there, but Maravon followed by a DMI or one of these products like Pristine or Maravon or Luna Sensation followed by a DMI. And that's to mix things up and actually give you different frac groups in that late season brown rot control. And so when you're looking at this, if you look at the Maravon followed by the Sevia, what you're going to see is the results are essentially the same as if you put out two applications of Maravon. That's what we've seen over and over and over again. That's good resistance management. You're using a lot of different frac groups. And then Miravis, which is the new chemical that's come on from Syngenta, again, it's very, very similar. It's SDHI, but it does have good brown rot control as well. So that's what you can get out of that. Now, the other thing that you can look at this and see, and this is one of the things I'm a little bit concerned about, and perhaps this is an artifact of being environment. Maybe we just really have really high resistance in the DMIs. But Sevia is a new product, just come on the market. In a lot of areas, Sevia is going to be better than other DMIs. In our case, we just don't really see that. When you look, especially down here at this particular row down here, if you're looking at Sevia or Indar, what you see there is by the 11 days after harvest, again, that's late, but you start to see a breakdown of that DMI and the activity. And we generally do see that these DMIs are not going to be as efficacious as what we would like for them to be relative to something like a Maravon or a Luna Sensation. Now, maybe that's just a standard. That also could be an artifact of the resistance that we have at the station. But nonetheless, there's some questions we have out of that. When you look at the data we have from 2021, you see essentially the same type of data. You look at your untreated up here at the top, again, a lot of brown rot, which is what you'd expect. The Maravon, again, if you put it out twice, I mean, it's a dynamo. But we can follow it with a DMI and we see really good response. So Maravon followed by Sevia, you get a really nice response. If you look at Luna, again, the Luna Sensation, Luna Flex, very similar to Maravon as far as the components in that particular product. Again, pretty good control that continues on. In blue, if you look at Sevia, this new product, Indar, and this is the highest rate of Indar we can put out. And then Inspire Super, which has another DMI, you can see that even at 7 days after harvest at that site, we're seeing a breakdown of the activity of those DMIs. Now, I think that could be related possibly to resistance, but we don't know that yet. We've not confirmed that. One of the things I want to look at this year and really kind of fine tune this a little bit more. When we're looking at these combinations where you put out Maravon followed by Sevia, the question that comes to your mind is Maravon just that strong? You know, in other words, if I just put out Maravon at 14 days prior to harvest and didn't do anything else, what would happen? So we're going to look at that and look at Pristine and some of these follow-ons as well. but we still want y'all to utilize these DMIs. If you look at it, again, every time we put out Maravon followed by Sevi in all these trials, we do well. It's just the DMIs by themselves. If you put those out back to back, especially if you have resistance, they're not going to do well for you. So there's a few questions that we have to tease out from what's going on there. But you can see I'm not satisfied with that DMI activity in those trials. That's what I'm saying. All right, let's go to scab and talk about some of the scab data. Of course, y'all are familiar with peach scab and what it does to us. I mean, it's a problem for us year in and year out if we don't spray well, but it's also one that we can control relatively well. And especially, we utilize the chlorothalonil in particular at petal fall and shuck split, and then sulfur and captan after that. And all of those products do not develop resistance. So again, those are good products for scab. They don't develop resistance. So we like for them to be incorporated a lot into our spray program. As far as the timing on when scab really takes off, it's going to be around petal fall, shuck split, and within a few weeks of that. If you look at where the most spores are being produced, it's in that time frame. So even by the time you hit shuck split, you've already had 25% of your scab spores have been produced. So there's a lot of sporulation going on during that bloom time frame at petal fall, and so there's immediately a cloud of spores basically ready to go onto that fruit and drip down onto it. So that's your time frame as far as when it's actually critical, and that's why petal fall and the shuck split time frames are so important. We've learned over time, again, if you look at what we call shuck split, we say shuck split's really critical. Well, shuck split's not one thing, and I'll reemphasize that again. You can see the distinction between some of the stages of the flower development in that particular picture and where shuck split is happening. It's very rare that you're going to have all of the shuck off at one time. If you get something like that where it looks like that, that's probably the exception, where you really see all these shucks off on those peaches simultaneously. That's why we recommend a petal fall and then also a shuck split application of chlorothalonil, because when you're at petal fall, what we call petal fall, you've already got a good bit of shuck split. And those little baby fruits are exposed, and that's, again, a critical time frame with a lot of spores. So you really want kind of a back-to-back, relatively rapid application of Bravo during petal fall and shuck split. And again, that little baby peach, that's the most susceptible time for not only scab, but also bacterial spot as well. And I'll mention that in a minute also. But again, that is a little baby peach that is just really, really susceptible to a lot of stuff. Okay, I'll let you take care of that so I don't really screw it up. All right, I'm back. You throw me off the stage in a minute, but not yet. All right. Talking about scab, I'm going to just go over a few things on scab. First of all, rainfall is critical during that time frame. If it's really dry during petal fall and shuck split, you're going to have less scab. I mean, that kind of goes with territory, but how often is it really dry during petal fall and shuck split? Going back to one of our dates from years ago, going back in 2013, you see this is a year in which we had a lot of rainfall. And I'm just going to concentrate on a few portions of this test. But if you look at the scab severity and the scab incidence we had in that particular trial, you go up here and look at your untreated. Of course, we had a lot. And here, just trying to determine differences between some of these treatments. You know, we don't do a full season program all the time to determine efficacy. But if you look at chlorothalonil at the top, Abound, or yellow jacket sulfur, you can see that sulfur is not as good as these other products. And I've told you all that many times, I think. The Abound and the Bravo are the two that are going to be most efficacious for petal fall and shuck split. Sulfur is just not as good. But when you look at the Bravo or Abound and you put them in combination with a full season sulfur program like you see down here, it's Bravo followed by the yellow jacket sulfur or Abound. They're very, very similar. So you could use Abound during that time frame, but again, we would discourage that because again, we want to keep that product for the brown rot at the end. So we want to use as much of the old stuff that doesn't develop resistance, but Abound does work also. Almost identical results, and we've shown that many, many times. The new product comes on the market, and this raises a lot of questions because this is so good, but Miravis has been brought on by Syngenta. And we first looked at this several years ago for scab when it was a numbered compound. And so it's really super, super exceptional for scab. And I'll talk to you about that in a second. It also is registered, as I've already told you, it's good on brown rot also. I mean, it's just a single SDHI, but it has really good activity. It's almost comparable to, if not comparable to, Maravon. So it's a really good SDHI, but it is a single frac code product. And it gives quite a few other things too. So it's a broad spectrum material that is out there, and scab's one of the things. Several years ago, back in 2016, Henry McLean with Syngenta actually came to us and asked us to look at this as a numbered compound because he knew what it did for scab and some other crops as well and other diseases. He knew what it did on pecan. And he said he thought it'd be pretty good on peach. And again, it was a numbered compound. So I had a student who went out and basically did a kind of a look-see trial. And yes, it had really good control. Now, we sprayed it several times in that year. I mean, you look at the number of applications we put out, I think we had 6 applications in that time, and we put it out a little bit late. We started late. So we know we missed some of the critical infections. So, but it looked good. In 2018, I've now gone back in and changed this, I think, from all the numbered compounds that we used to have in these slides. But you can see that looking at just again, here we have 3 applications of the products, Petafall, Shut Split, and then a cover spray. But when you look at Miravis in particular, or combinations of Miravis where there's some other products involved, you can see that that is actually a pretty active compound by comparison to Bravo in those early applications. So it is a better product as far as comparing it to Bravo. And when you look at the severity in particular, I mean, just 3 applications, I mean, you would not downgrade that fruit. I mean, that was it. We put it out early season, it's done. And so that's really tempting to figure out how to use that in a different way. This is 2019 data. Here you're looking at, again, I don't want you to concentrate on Miravis because that's the one that's registered for us. But this, not as much scab, though. It was a drier year. But here's your Bravo. We had 4 applications in this case. We're comparing to the Miravis. And then this is a full season program. What I'm pointing out here is even with a full season program with 2, or really three Bravos, we had, or 4. We actually put out a lot more Bravo that year. Is the reason we had a long delayed period of time for shuck split. But we put out a lot more Bravo and Yellow Jacket Sulfur, and we still had some scab. And with the Miravis, we just didn't have any. I mean, it was the cleanest fruit I've ever seen. So you say, well, yeah, that product really is more active. It really does have this longevity associated with it. It brings us to 2021, and actually Brett had a graduate student who wanted to do a special problem to get some credit for classwork. And I said, well, I got a project for you. So we put her to work on this. And so this is the rainfall from up in the Watkinsville site where we did this. And you can see we had plenty of rain. And right around Petafall and Shuck Split, we had a lot of rain too. So we had a good condition to have some scab and to do a trial. And these are the things we tested. So I'll stay on this for a second just to kind of show you what we're looking at here. Again, we're not putting out a lot of applications. We're only putting out Petafall and Shuck Split applications, or Shuck Split and Petafall, or various combinations. Here's your untreated. Again, the incidence is pretty high. Not huge scab severity, but certainly enough for us to test this concept-wise. Here's Cap 10, Petafall and Shuck Split, Abound Petafall and Shuck Split, and Bravo Petafall and Shuck Split. And this is what you get out of that. Okay, that's still, that would downgrade you on scab if you did that. And it shows they're active, but it would downgrade you at the end of the season. When you go down here with 2 applications of the Miravis, we're down to a point where you probably would not be downgraded. You know, on average, you're seeing 2 or less spots per fruit on about a fourth of the fruit, 28.8%. By comparison, these have more than twice that amount of fruit. So again, this product is just that much more active. It's very, very active. We also looked at even just a single application of these, and again, you get really good response with the Miravis and single applications, and then some. We played around with Bravo and the Miravis, or Bravo followed by Miravis. Again, this is incorporating it. If you were to incorporate, what I'd probably do is I'd put out Bravo at Petafall and Miravis at Shuck Split as far as resistance management, if I did this at all. And again, there's a danger there associated with using Miravis at that time frame. So the conclusion out of all that is to say that this is a new product. It's very active on scab. There's a temptation there for, I don't know if your mom ever told you this or not. Just because you can do something doesn't mean you should do it. And you can do this, okay? So you could use mirrors, and there may be a time when you need to do that. If you're having problems with scab, I mean, if there's a reason that you're having problems with scab, and you want to help to knock in the head, Miravis is certainly an active product. If you know going into the pedal fall shuck split time frame in particular, you're going to have a lot of rainfall. If the prediction is rain and more rain, this might be a time where you want to use that at the shuck split application, just because it is systemic. Bravo, the only time I've seen Bravo quote unquote fail for me in my trials is when I followed it immediately with rainfall. And we have had what I would call a true failure then. But if you see that you're getting into a situation where you say, well, I might need some extra help on scab, then I would potentially put out Bravo at pedal fall and then follow that with this other product, Miravis at shuck split. And that's a possible time you could use it. Again, I will say this over and over because it is one of these products that we want to save that whole category for brown rot. I would not overuse this. I try to avoid it if at all possible, but it's another tool in our chest. It is there and it's really good. So it's something you need to be aware of. And then, of course, we can use it also at the end of the season as another material, but it is a single SDHI product, whereas something like Maravon is actually a dual product with a SDHI and a QOI. So for resistance management, it's actually really, really good. And of course, we have the other products that are all thrown in there. But that's where we are with that. So I just want to bring it to your attention. You know it's better as far as scab management. It's a real good product. And you just need to figure out when you're going to use it, if you're going to use it, with the risk associated with it for resistance development. So just to reiterate this, Miravis is comparable to Maravon in brown rot efficacy. Very good product for brown rot. Sebia is just at this stage, I just say it's another really good DMI. I don't think it's any different than Endar at the high rate. We've not seen it really separate out. It does separate out in some other commodities I work on. It's actually much better for powder mildew, for example, in grape than other DMIs. But on peach for brown rot, we just haven't seen it separate out as being better. It is a good new DMI. And then the scab activity for Miravis is just real tempting to use it when you need it in the early season. Okay, so if you think you need it in your program, it's out there and it is really good for scab. So that's all I'll say about that. Let's go to bacterial spot real quick, just to talk about some of what we've done this year. HaHa has set up these weather stations with us down in Georgia and throughout South Carolina. This is kind of a teaser on some of the stuff that we'll be seeing. This is an example of some of the data that you get off these stations. It can give you more data and possibly more than you can understand. But I am excited about what we're going to get out of this, understanding bacterial spot better. The first thing I can say is we had 3 sites, and you can see we had also 3 different times we took leaf samples and where we could fruit samples. The thing I got out of it this year, if you look at rainfall, we did have some dry periods in here right before we started sampling, and this is about where you're going to be at the stage where the fruit should be getting a little bit more resistant to the bacterial spot, pit hardening type stage. But it was dry during that time frame, and then it got wet. But look at the leaf wetness. What I got out of this is leaf wetness, at least from the standpoint of what it's monitoring. It says we have a lot more leaf wetness almost every day when it's raining a little bit than we think we do. And leaf wetness is probably going to drive that system. The rainfall is important, but leaf wetness is even more or as much of importance. Here's another example of that in the second site and the third site. So I think in the end, we'll start to get some information that's going to help us to understand what the disease parameters are for bacterial spot a little bit better. One of the things I've already started to get out of this is it questions a lot of things. I've shown this slide many times. This is old data. It's coming in from Ron Gatitis and Paul Bertram from back in the 80s. And this is just on leaf bacterial spot, but when they've inoculated at different times, what it says is, or seems to say, is that if you're getting late inoculations with bacteria where they put them in on the leaves, you don't get a lot of development after that point. So they're putting out at different stages and seeing what happens. I start to question that after this year. because we're looking again, these are the three locations we were at, and this is looking at the number of infected leaves per shoot. And these heat maps, you can see that without regard, we see an increase in disease on the leaves after that time frame we started looking. And so all three of those cases, it says, well, that was late. And we started out with very, very little, and it did get worse. And so I started to question some of that older data. That data was never published anywhere except in an abstract, but it's the best data we have. We're going to get better data out of this than we've had in the past, just by looking at it more intensely. Again, you're looking at percent infected leaves per shoot. Again, they are increasing. So leaves, that's one thing. All right, well, you're worried about leaves because you don't want to defoliate your tree. It's not a good thing to defoliate the tree from bacterial spot. But what you're really going to be worried about, again, this is saying the same thing. You just see that leaf damage does increase over time. Whoops, wait a second. That's the leaf damage incidence. Oh yeah, okay, I'm sorry. I thought I skipped ahead. So let's talk about the fruit infection. That's the next thing. So there's also good information, again, but in an abstract. I don't think it's ever published anywhere else. But it says that yes, and we know this, and we believe this, the early season is the time frame in which bacterial spots coming in the most. But we've kind of had this dogma out there, more based on observation. When you get to pit hardening, you're going to be relatively safe. And I think that is true. You're relatively safe. But I don't think we can say that you're not getting any infection after pit hardening. And I think that's been kind of a standard, kind of a urban myth, if you want to call it, that's out there. And this is another statement similar to that when you're in dry years, with little or no bacterial spot observed on fruit at 6 to 8 weeks after such split, fruit infection is likely to be limited. And I think that is true, and especially if you have dry conditions to harvest, because we see that. And again, pit hardening may be a good estimate of about when you really start to get in a little bit more fruit resistance to the bacterial spot. But this year, again, we started out with virtually no bacterial spot on fruit. We only had 2 locations with fruit. The other one was frozen out and there was no fruit there at all. But when you look at fruit infections, again, we start out in June and we have virtually none. And then by the end of the season, we have a pretty good incidence of bacterial spot on the fruit in those locations. So I said, yeah, it did increase. Okay, so that's something I don't know we can go back and say that anymore. Again, just looking at the bacterial fruit damage itself, that's another way of looking at it. Just saying, yeah, it really did go up. So it wasn't that because we had virtually none and it did go up. We did have some rain. So when you look at something like this, where you have early season infection, especially prior to that pit hardening, you can expect to see that in the season. I think that's just going to be something we can say, yeah, of course it's going to do that. That's no surprise. This has been the thing that I hope that some of this research will help us to interpret is when you see the spotting like that, is that all late season infections or is that latent infections that are just showing up late season? I tend to think now more that this is actually a late season infection. I think we are just getting some light infections that are going to continue even after that pit hardening stage. All that's important for us to understand how to manage this disease post pit hardening. You know, we need to keep putting out that copper. We need to keep putting out the antibiotic. Something even at that stage just to keep that from happening. I think this research will help us to really fine tune that answer to that question. So the conclusions on that, one thing I can see right now, I don't know exactly how to interpret it. I don't know here yet. I think she will be speaking at some point. But I think there's a lot more leaf wetting than we give ourselves credit for. I mean, I think it's out there even when we think it's relatively dry. There's some leaf wetting at night or in the morning. And then the leaf and the fruit bacterial spot, even after we get into the summer, does increase, especially if we have enough rainfall. So I do think those are some things that we're going to really tease out of this a little bit more so we can give you better information with that grant. Had a lot of students working in the summer. They counted the bacterial spot till they basically were ready to shoot each other, I think. But nonetheless, I appreciate their help. A lot of effort to count all those spots. And with that, I will, and if y'all have questions, I'll try to answer. The only time I do it, I'm sorry, I'm not cutting you off. Whether is that your question? Should we do that? Yeah, yeah. I'd say that the way I would look at it is it's a tool in your toolbox. You know it's there. You know it's good. The risk side of it is I'm using an SDHI early in the season that could develop resistance to brown rot, and brown rot's out there at the same time. I don't want to see you doing that often. And so what I would say is if it's dry, in particular, if it's dry during petal fall and shuck split, 2 applications of Bravo should carry you through. Are you having problems? I'd ask the question, are you having problems with scab? If you're not having problems with scab, I wouldn't change anything. It falls in the same category. I've told you also, I don't know if y'all remember or not, you can put out Bravo with your oil applications in the wintertime too, and you can reduce scab. So, but if you don't need it, don't do it. I do occasionally have producers that have a real hard time for whatever reason controlling scab. And I'd say until you get it under control, get the number, your total tree scab levels down, you can use these tools to help you get it down. But I wouldn't do it unless you had to. Now, I'd say that if I was a producer, for me personally, if I saw a lot of rain coming during that petal fall or shuck split time frame, I would be tempted to put out the Mirabus once at the shuck split in those years where it looks like it's going to rain a lot, because I do know that Bravo will wash off. Yes. Yeah. Yeah. 3 weeks is a long time. But I do think we can capitalize on dryness. And so I guess what I'm trying to say is when it's raining a lot, yes, you do need to tighten that schedule. And I'd say probably you have something going as much as possible weekly or back your spot when it's raining. I really think you have to. And you can alternate copper, like you said, would be a good way to do that. Yeah. I think the problem that you guys run into a lot of times it's hard to get over everything, especially if it is wet. Because you actually slows it down as far as getting over your total age. And so you might not be able to tighten it that much, but tighten it as much as you can when it's wet. As far as your Oh, I know, I know. Yeah. If you're alternating the copper with the oxygen, I think it's unlikely you're going to overdo it. Where I've seen what I call substantial damages in our trials is where we're going in weekly with copper back to back to back copper, and it will build up. And so, especially if it's dry, it really builds up. And we have induced enough damage with copper that we've probably taken off more leaves than we would have with natural spot. But it does help the fruit. So it does. If you don't, you may kill your tree, but your fruit look good. That well, you have to have some. Yeah, you got to see some damage with copper, or you're not getting enough out of it. But it does build up. Yeah, thank you. Mhm. Oh, right.
Yeah. Next we have with Dr. Guido Bravo, plant pathologist from the University of Talk to us about anthracnose.
So thank you very much for having me, and thank you very much for allowing me to talk about anthracnose disease of peach. Uh, doesn't happen very often that we need to have a talk on this topic because it is not really an issue that uh, we find important every year. But every once in a while, it does flare up, and it can actually cause economic damage. Anthracnose disease is common in other crops as well. So anthracnose is caused by Colletotricum species. And yes, it's a minor disease issue in peach, but it's a major disease issue on apples, on grapes, and on strawberries. And some of you also grow strawberries specifically as a diversifying crop. And you all know that anthracnose in strawberries can be devastating. And so the talk today will relate not only to peach, but also a little bit to strawberries as well. Um, there were a few outbreaks with significant economic damage in South Carolina and in Georgia. So in South Carolina in 2004, I saw it first in Spartanburg County on baby gold peaches. This particular grower worked with Gerber, and Gerber wanted to have peaches that were tree ripe with lots of sugar, and that's where we saw a lot of summer diseases, and one of them was anthracnose. In 2014, 2015, in Saluda and Chesterfield counties, there were significant outbreaks with economic damage, and that was associated with fungicide resistance development, specifically resistance development to the FRAC 11s. Those are our QOI fungicides. I'm going to talk about this a little bit more later. And in 2021, we had another outbreak in Chesterfield County, and that was associated with a nearby strawberry field that was having some anthracnose issues, and we believe that the disease moved over to the adjacent peaches. Now in Georgia, there was a major outbreak in 2021, and that's probably the reason why I was invited to give this talk. Now, in talking to Phil Brennan, yes, it's there, but typically it doesn't cause economic damage. At least it hasn't in the last 20, 25 years maybe. But I did find some references in the literature that in 47, 48, and 49, there were significant outbreaks in Georgia, and that was associated with nearby blue lupine cover crops. Blue lupines are highly susceptible to anthracnose as well. So what happened in 2021? I'm not sure. I didn't sample fungi, but I suspect that we may have experienced a resistance issue. There are basically two major families of pathogens that can cause anthracnose disease of peach and also of other crops. One is Colletotricum gliosporides, short the GLOW, and the other one is Colletotricum acutatum. Those are two families of species. Actually, there's some subspecies belonging to these families, which relates to the title of the talk. It's complicated. Colletotricum gliosporides, the biology and epidemiology is different from that of Colletotricum acutatum. Specifically, it does much better under hot conditions. 30 degrees Celsius, it does very well, whereas Colletotricum acutatum does very well a little bit lower temperature at about 25 degrees Celsius. Glowis varieties, or the glow, is best known to infect vegetative tissue, especially strawberries. Now that is the one species complex that produces both sexual spores and non-sexual spores. So that's something to keep in mind. So it has a greater ability to adapt to environmental stresses, including the stress of a fungicide application. And it mainly overwinters on dead wood, on plant debris, and on alternate hosts. Now, with regard to Calatachicum acutatum, maybe I can use my cursor here to point a little bit. You see this? Yeah. So with regard to acutatum, so it does very well on at little bit lower temperatures. It is best known to affect the fruit. Now I can't see my cursor anymore. Disappeared. That's just very hard to see. Anyway, the angle is just not very good. So it is known to affect the fruit a lot, especially apples, grapes, peaches as well. But it only is known to produce asexual spores, which is good in a way because genetic diversity is not as great, and it also mainly overwinters on mummified fruit, infected twigs, and with regard to strawberries on symptomless transplants. Now, the other significant information to keep in mind here, the glow versus the acutatum, those two different types of species, complexes, they also vary in their susceptibility to fungicides. For example, the Glowis varieties is much less susceptible to fludioxonil, which is a mainstay in strawberry production, but can also be used post-house on peach production. On the other hand, it is highly susceptible to Topsin M, much more susceptible so than Calatachicum acutatum. So there's these differences in fungicide susceptibility that we need to be aware of. So knowing your species actually does matter. So before I came to Clemson University, Eldon Zaire was the man in charge for plant pathology issues, and he did a study with Mr. Bernstein. I think it was a Mr., unless it was a Mrs. I'm not sure. But Bernstein and Zaire published a paper that showed that isolates from peach collected in Georgia, in the upstate of South Carolina, and also in North Carolina were primarily Calatachicum acutatum, whereas there were a few isolates collected south of Aiken that were Calatachicum glowis varieties. So both species were present, but the major species that they dealt with in the 1990s was Calatachicum acutatum. And again, it does matter because of all the biology differences and the fungicide susceptibility issues. But then we've had those outbreaks in 2014 and 2015. And my postdoc and I, at the time, we were going out and collecting samples from all these affected areas in the upstate, in Chesterfield County on the ridge. And we found that the overwhelming species was actually Calatachicum glowis varieties, the glow. So it had changed from acutatum to the glow. Why? That's a good question, and there are probably several possible explanations. One is that the glow was suppressed by FRAC1s, such as Benumil and Topsin M in the 1980s and 1990s. The other possibility is that acutatum has been suppressed by the FRAC11s since the year 2000, when pristine was introduced, and then following the introduction of pristine, Merivon, Luna Sensation, and Abound were used. And acutatum is just much more susceptible to those kinds of fungicides. Another reason is the fungicide resistance development. So because the glow we talked about sexual and asexual reproduction. The glow produces more genetic diversity. It might be that it just happened to develop resistance much better, and it outcompeted Colletoticum acutatum. And the fourth option is climate change. Glosporides is more suitable for hotter environments. It can survive hotter environments a little bit better. So what does Colletoticum glosporides look like in culture? Those are the isolates that we collected from South Carolina in 2014 and 2015. And you can see that the culture morphology is very different. You can see on the bottom cultures that look grayish and white, and on the top cultures that are fluffy, white, and a little bit of pink in them. And what this basically is we put a single spore of the fungus in the center, and we let that single spore grow for about 4 days, and then within 4 days it covers an area that's about this big. So this is what that is. But it shows you that there were different phenotypes, and they actually corresponded to different species. So within that glow, we had different species that also have their own little biology and their own fungicide sensitivity. So again, complicated. This is what the fungus looks like on peach. On the left is artificially inoculated peach that we handled in the lab. And you can see those lesions that at first look a little bit like brown rot, but brown rot, for example, it doesn't change the morphology of the peach. Anthracnose will cause a lesion that is sunken, sort of dips into the fruit and produces either a gray fuzzy mycelium on top, or it produces even pink spores. On the right-hand side, those are pictures from 2021 from Chesterfield County, and that was an outbreak that we recorded last year. So I already mentioned a little bit the different genetic compositions of the species. What we found in 2024, 2020, 2014, and 2015 in South Carolina were several different genotypes depending on where we isolated. So in Chesterfield County, for example, we found the red genotype that was closely related but not identical to the blue genotype over here found on the ridge. Found a purple genotype down here south of Aiken, and another genotype up here in Oconee County. And these genotypes were characterized by the nucleotide sequence of 3 different genes. So that is interesting, but maybe not relevant for us. But in this case, it is, because those genotypes also correlated or corresponded to fungicide sensitivity differences. So these genotypes over here, the red and the blue and the purple, those were all completely resistant to MBC and QI fungicides. Those are the FRAC 1s, Topsin M, and the FRAC 11s, your Pristine and your Maribon, your Luna Sensations. Completely resistant because of point mutations in the target genes. And because of those point mutations, the resistance is inheritable and it's very steady, and there is no known fitness penalty to those resistances. So it's something that we really need to deal with. So that was a major discovery, and it could be that the 2021 outbreak in Georgia was similar to that, but I don't know. I haven't studied those isolates. Maybe something we can do together, Phil, next year if that happens again. So just to make sure that you understand this, those genotypes are resistant to Topsin M and to the QI fungicides such as Pristine. So Pristine, Luna Sensation, Maribon, Dr. Brandon has already talked about those. Those are essential for brown rot control. Those are being used early in the pre-harvest or during pre-harvest season. And they consist of two different active ingredients. One of them is a frac 7, and the other one is the frac 11. Over here, frac 7s and the frac 11s. So if your anthracnose fungus is resistant to frac 11s, that leaves the frac 7s as the only active ingredients that would do anything for your anthracnose management. And that is at a time when the fruit ripens and when the fruit is becoming increasingly susceptible to anthracnose disease. So the question is, can we count on the frac 7s to actually have any activity against those modified anthracnose fungi? And the answer is no. None of the frac 7s that are currently available to us in those three compounds have any effect on the anthracnose disease. The only frac 7 that is actually having an effect against Calotropis species, Eprovia. The active ingredient is benzobindiflopyr, but that one is not registered for peach, and it will not be registered for peach according to the company. So again, how does that relate to our spray program? And Dr. Brennan has already mentioned that we have a really solid spray program that we developed over the years that we should not mess with, and that actually works really well against all the diseases that we encounter, including anthracnose if there is no resistance developed. So the bloom sprays over here, they're effective against blossom blight. Now we've got your petal fall sprays. In the cover sprays, we apply Captan very often. Captan is an excellent product that keeps the anthracnose infections at bay, and it will keep the anthracnose levels very low. And then we go to the pre-harvest season where we spray our first Marivon or Pristine or Luna Sensation. That's the critical time right here that we need to address. And again, this is a program that if we didn't have any resistance, it would work great, and it has worked great in the past for anthracnose and brown rot management. So what are we going to do? So if there is no effect of those frac 711s against anthracnose, what are the options? The frac 1s are no options because most likely we are seeing resistance to those as well. Roveral can only be applied early season. It's not very effective either. The frac 7s are out, the frac 11s are out, and the frac 12s can only be applied post-harvest. So what's left are Captan, and definitely Captan is something you should be using if you have anthracnose problems, but you can't use it pre-harvest. Then there's the frac 3s and the frac 9s. So let's take a look at the frac 3s. Maybe there's something there that we can take advantage of. There are several frac 3s available for us, for fruit growers, for peach growers. And those are mefentrifluconazole, difenoconazole, and so on. Those are the active ingredients of trade names Sevilla, Inspire, Super, Bumper, Elite, Rally, Indar, and Top Guard. So those are available. And you scan the literature for activity, you find so-called EC50 values that were published. The lower this EC50 value, which indicates an in vitro inhibition, the dose at which fungus is 50% inhibited in vitro. The lower this number, the more active the active ingredient. So you've got a 0.4 here for mefentrifluconazole, for example, a 0.5 for difenoconazole. So you got some good ones right here, and then you've got some not so good ones right here. And the same is true, so that's for the glow. And for Calotropis ectotatum, it's very similar. You've got a few DMI fungicides that sit in front of the boat, and you've got a few DMI fungicides that are sitting in the back of the boat. Now I indicated those in red, those numbers over here, because for mefentrifluconazole or Sevilla, we just don't have a lot of data. There's only one study out here that shows a very low EC50 value. There's 3 studies out here that show low EC50 values for this species. Yes, I rank them 1, but we have to be careful, just not enough data out there. There's much more data out there for difenoconazole and propiconazole because they've been around for a longer period of time. All right, so to just review this, we've still got Captan, we've got maybe the FRAC3s, mefentrafluconazole seems to have pretty good activity, difenoconazole, propiconazole. Now, some studies do show that the FRAC9s, like pyrimethamine, Vanguard, and cyprodenol, which is a component of Inspire Super, also have activity, but it's really dependent on the species. Some species are completely resistant to FRAC9s, and other species are highly effective, highly susceptible. So it depends really on the species. Now, I highlighted Inspire Super because there are some studies out there that we could pull to verify those in vitro data, those in vitro efficacy data. So I looked at those and came up with about 7 studies that I was able to analyze with regard to efficacy. So those are studies that were done on apples, bitter rot of apples. Bitter rot is caused by Calototrichum clostridium and Calototrichum acutatum species. It's a mixture, very hard to control. Just ask any apple growers in North Carolina. It's a very hard to control disease, and if something works against bitter rot, it ought to work for peach as well against anthracnose. The results show, though, and those are studies that I collect or that were conducted between 2015 and 2017. The results show that Inspire Super applied at 12 fluid ounces. There were mixed results. In some studies, the effect was 100%, and in other studies, the effect was 0%. 100%, 0%, and then some other studies, mixture, 25% efficacy, 66% efficacy, 75% efficacy. So what does that mean? Is it, Russian roulette, or what is this? What does it mean? If you dig a little deeper into the data, you will find that the efficacy is directly related to disease pressure. The higher the disease pressure, the lower the efficacy. So those are all the studies with high disease pressure, and they didn't show much efficacy. Those are the studies with low disease pressure, and the efficacy was pretty high. In other words, I think if we were to use Inspire Super, which has difenoconazole and cyprodenol as active ingredients, it needs a booster. We need a booster for Inspire Super. And there's actually some evidence that propiconazole, which we know has pretty good EC50 values against Calototrichum species, that propiconazole and difenoconazole, which is in Inspire Super, that they have synergistic interactions. The reason is molecular based. There's 2 different enzymes that need to be controlled in Calototrichum species. One of those enzymes is better controlled by difenoconazole, and the other enzyme is better controlled by propiconazole. But together, at least in some controlled studies, it shows that there's some synergistic interaction. That plus DMIs are known to work better when the dose is increased. And that is also true for anthracnose. So if you have more DMIs in our tank mix, higher dose, and in this case, 2 different DMIs that have synergistic interaction, it should work really well. How can we integrate this into our existing spray program? Here's a suggested spray program for orchards with brown rot and anthracnose problems linked to fractal resistance in Calototrichum species. Again, if you don't have anthracnose problems, you probably don't have any resistance. And the original spray program that Dr. Phil Brandon showed and that I showed in the beginning are just going to be fine. But if you have an exceptional situation with resistance concerns, this is what you can do. You can integrate your Inspire Super plus the booster plus bumper, tilt, or Amtide, all those trade names that have propiconazole as active ingredient. So you can use that mixture maybe at 21 days before harvest and then at 7 days before harvest, intermixed with your FRAC 711 mixture, and that should give you at least some control of those resistant strains. That's the best guess that I have for you. And unfortunately, I don't really have data to back this up because anthracnose is just not something that I have in my experimental orchards. But it's something that you should try, and it should give you some good results. So in summary, multiple species of Colotochicum can cause anthracnose of peach, and the composition has changed over time. We moved from Colotochicum equitatum to Colotochicum clostrides. Anthracnose is typically a minor disease in South Carolina and Georgia, but has caused problems in the past when either a new alternate host was introduced or when the pathogen had developed resistance to the FRAC 11s. The current spray recommendations are not effective against anthracnose if the pathogen is resistant to FRAC 11s. And managing FRAC 11 resistance could be accomplished by integrating Inspire Super plus a booster, which is a Amtide, bumper, tilt, with current recommendations. But again, solid evidence data is not yet available. That's just to acknowledge some of the sponsors of these research projects. Thank you very much for listening, and I'm available for questions. Yes. That is true. That is something that the competitors especially in those orchards where we have this problem. That would be a great addition to this site.
Okay, is that it? Thank you, Dr. Schnabel.
Yep, I'll get for you.
Thank you. Okay, next we have Dr. Libby Sinwitz with Clemson University Virology.
Okay. Thank you, Andy. Thank you all for having me today. So I'm going to give an update on the Clemson Clean Plant Center and the three aspects of our program, which are the Southeastern Budwood Program, our diagnostics and soon virus elimination therapy capabilities, and then our Prunus Foundation that we maintain at the Musser Farm. So I want to start off just by saying that all of this work that I'm going to be talking about today is conducted under the auspices of the National Clean Plant Network. And this is a USDA program, now permanent USDA program that is, the whole mission of it is to provide a source of asexually propagated plant material. So there are several crops, which I'll mention on the next slide. But just to ensure the global competitiveness of the specialty crop producers and protect the environment with a focus on viruses and other graft transmissible pathogens. So the National Clean Plant Network, here's a map showing some of the different clean plant centers across the United States. And you can see the different crops are listed there on the left side of that figure. And so we have our fruit tree center in Clemson, and there are two other fruit tree centers, one at Washington State University in Prosser, Washington. That's called the Clean Plant Center Northwest, and I'll refer to them a little later in this presentation. And then the other one is at UC Davis, and that's called Foundation Plant Services. The National Clean Plant Network, or NCPN, was implemented in 2009 as part of the 2008 Farm Bill. Now, as I mentioned, it's a permanent program, and it has involvement from all of the different arms of the USDA, and essentially it supports clean plant centers throughout the United States with annual funding, but also kind of provides this network of similar centers across the U.S. that are doing similar work, and we can kind of collaborate and not duplicate efforts too much. So the crops that are covered, fruit trees and grapes were the two initial crops in the NCPN. And since then, we've added citrus, berries, hops, roses, and sweet potatoes. And there's always a few more crops that are kind of knocking on the door to get into the NCPN. I mentioned where the fruit tree centers are, and NCPN is kind of a unique thing because we have members that are researchers, so those of us who are located at the universities. We have both federal and state regulators that are part of these meetings, and we have growers and nurseries. So it's not often that you get all of those people in the same room working towards a common goal. So I just want to start off and review kind of virus prevention in the first place, and then some of the major viruses that we're focusing on. So why is virus prevention important? First of all, just knowing that once a tree or a plant is infected with a virus, then it will remain infected for the rest of its life. So we've really only got one shot to prevent a plant from becoming infected. So all of our management tactics are based on prevention. And of course, viruses can cause damage to orchards, whether that be in the form of reduced fruit yield, fruit quality, weakening of the trees, and potentially interaction with other pathogens. And then this can result in, of course, economic losses, and that can be in the form of the cost of tree removal and replacement, waiting for the new orchards to become productive, and of course, weakened trees can result in lost revenue. So that's why viruses are important, and so which viruses are important to us here in the Southeast. And I say viruses, but I also am referring to a couple of other graft transmissible pathogens. So the major graft transmissible diseases of peach in the Southeast are peach stunt disease, which is actually a synergistic interaction between two viruses, Frühendorf virus and Prunus necrotic ring spot virus. And the disease severity varies by cultivar, and you can see some photos here by my predecessor at Clemson, Dr. Simon Scott. And then the other right now is phony peach disease, and this is caused by Xylella fastidiosa, and it's resurging in some areas, mostly a little further south. And this is vectored by sharpshooter leafhoppers. We are doing some work currently in collaboration with Phil Brannon and his PhD student Kendall, and we're working on trying to see if this is graft transmissible and how graft transmissible it is. What's the efficiency of that? So Frühendorf virus is graft and pollen transmitted. It's widespread globally in stone fruits. It causes various symptoms, but they could include stunting, shortened internodes, which is what's pictured here, and gumming and bark splitting. And we do have some preliminary evidence of increased susceptibility to bacterial spot in PDV infected trees, and this is some ongoing work in collaboration with HaHa at the Edisto Rec. The other one, I kind of pair these together. Prunus necrotic ring spot virus is graft and pollen transmitted, also very widespread in stone fruits globally. And the symptoms of Prunus necrotic ring spot virus are often characterized as initial shock symptoms, so early in the tree's life. These are some photos from the Musser farm where this is in the variety block there, and you can see that the two first trees there were PNRSV infected, and they're a lot smaller. So those are what we're calling shock symptoms. Sometimes you'll see the shot holing, but these symptoms are by no means diagnostic of PNRSV. And sometimes the trees will even, seem to recover as time goes on, but the virus is still replicating and moving throughout that plant. Plumppox virus is a very bad word. This one has not been detected in the southeast, but we have to remain vigilant. This is a podovirus that's vectored by aphids. Of course, it was detected in Pennsylvania in 1999, and then in Michigan and in New York. We also know that it's in Ontario, Canada. Now it's officially, according to the USDA, eradicated from the United States. And it's extremely devastating. There are very harsh eradication procedures that take place if we do find Plumppox virus. And this is one of the few examples of a virus that can actually kill the tree. I've added in tomato ring spot virus here. We haven't really seen this much in peach production in the southeast. I'm not sure if that's just because we aren't looking for it or if it's really not very prevalent. But this is a nematode vectored virus. It's vectored by dagger nematodes. It can persist in weeds and in nematode populations in the soils. There's a very wide host range, both the nematode vector and the virus do. And it can cause thickened necrotic bark, stem pitting, and decline of the orchard over time. I mentioned this one because recently we've been doing some virus surveys in blackberries too, and we find tomato ring spot virus a lot in blackberries. So we're not really sure what's going on and if it's prevalent in peaches. And if not, then I'm curious why. I also mentioned this one. We don't really see X disease phytoplasma in the southeast, but right now it is causing major problems in the Pacific Northwest, in Washington and Oregon. These are some photos from Dr. Scott Harper at Washington State. And they're having a lot of issues up there with phytoplasmas, not just Western X, but some other ones too, and a couple other viruses. That mostly they're worried about it in cherries, but some of these pathogens can also go to peaches. So just a note that we should be vigilant of these things that are going on in other parts of the country. And really, viruses should be easily preventable, but they can quickly spread if the inoculum is high. So these are two examples of peach blocks in South Carolina. One is a heavily infected peach block that's infected with PDV. So that's what all of the green is there on the left. And then what we see, more commonly are these sporadic virus infections like are shown in the block on the right. But if you do have a block like this heavily PDV infected one, the nearby blocks are much more susceptible to being infected with PDV through that pollen-borne spread. So the first kind of major activity that we have at the Clemson Clean Plant Center is the Southeastern Budwood program, the virus diagnostics. So essentially the way this program works is that we establish blocks of anywhere from 50 to 100 trees per cultivar. These are established on farms in South Carolina and Georgia, and we collect leaf samples from these in the spring, and we test for three viruses by ELISA. So we test for PDV and PNRSV, the two pollen-borne ones, and then also Plumppox virus, which again has never been found in the southeast, but we're always on the lookout for it. And then once we do all of the testing for these blocks, then we put together some maps, and we provide these maps to the participating nurseries, which can then use them to collect budwood from these trees that are negative for those three viruses. So this program was started about 20 years ago by, as a collaboration between the growers in South Carolina and Georgia, led by the Peach Councils of South Carolina and Georgia, and then my predecessor, Dr. Simon Scott, and the nurseries, which are based in Tennessee. And this program has resulted in overall lower virus incidence in South Carolina and Georgia over the last 2 decades. So these are just the results of the Budwood program each year. So this is not, a very scientific survey, but it's the best data that we have on overall incidence across those 2 states over time. So this program overall has been successful. And, of course, we still find those viruses every year in those areas, but the goal is to, reduce that as much as possible. In 2021, we tested 38 different peach cultivars, and These were, again, across South Carolina and Georgia. This amounted to exactly 2,700 trees, which was amazing that it worked out that way. And then what we found was that 3.6% of those trees were infected with prunus necrotic ring spot virus, and 0.6% of them were infected with PDV. Thankfully, we've escaped PPV for another year, and we didn't find that at all. So, I want to kind of go into the definition of a clean plant. I think there's a lot of confusion out there about the results that we get from the Budwood program, and referring to a clean plant. So what is a clean plant? The NCPN centers define a clean plant as a G1 or generation 1 plant. So that's where our foundations come in. And then from these nuclear stock materials maintained at foundations, then we can produce the next generations of plants. So G2 would be like nursery mother blocks, and G3 and G4 would then be production plants that are delivered to the grower. And some states have certification programs through the Departments of Agriculture that regulate at the G2 and the G3 and G4 level. Not every state has a certification program like this. We do not have one in the Southeast. I want to reiterate that the Southeastern Budwood Program trees are negative for 3 viruses, PDV, PPV, and PNRSV. But the clean plants, as we define them, are negative for all viruses that are known to cause damage. So this to give a little more information about the G1 through G4 certification model that's used in some other states. Again, G1 are those foundation trees that are maintained at clean plant centers. They're rigorously tested for graft transmissible pathogens, and they're maintained under conditions that minimize the risk for reinfection. The G2 would then be nursery mother blocks, and then G3 would be increased blocks at nurseries, and G4 would eventually be those what we refer to as certified plants that are delivered to the grower. Of course, with these different programs, there are different regulations. For example, required testing and visual inspections that occur at each of these stages. The G2, G3 level can be blurred depending on the state and the production system. But the key here is that all of the certified material in these programs originates at foundations that are maintained at clean plant centers. Again, tested for all known viruses that cause damage, and kept at the highest sanitary standards, and they are true to type. So that kind of brings us to the second part of what we're doing at the Clean Plant Center, which is diagnostics for the whole prunus panel, and eventually virus elimination therapy. So we do some advanced diagnostics for a panel of 26 viruses, viroids, and a couple of graft transmissible bacteria using some advanced techniques like high throughput sequencing and PCR. And then if those accessions are found to be infected with a virus, then they can be subjected to virus elimination therapies. And this is a combination of heat therapy or thermotherapy and tissue culture. So overall, this diagnostic process will target these harmful and economically important pathogens at the optimal times of detection, and we have to test multiple times before officially declaring a plant to be clean. And so you can see in this figure here on the right that the new plant accession candidates would be at some places they can do the foreign imports. So we don't do that at Clemson, but you can import new peach varieties through the Clean Plant Center Northwest at Washington State or Foundation Plant Services at UC Davis. They both have the imports for that, or they both have the permits for that. And then domestic selections, which is a goal of ours to be able to do in the next couple of years, and new cultivars. So things coming out of breeding programs in the U.S. So we can subject those trees to disease and pathogen testing. Of course, if they test positive, then it goes into the pipeline for the virus elimination therapy, and there's a series of retesting until we can officially declare the plant clean. And then once it is clean, then it is destined for foundation. So then we'll maintain it under these conditions that minimize the risk of reinfection, and we do annual testing on those. And then the foundation plant stock can then be propagated out to nurseries. So the prunus panel, dive in a little deeper here. These are the diagnostics for the prunus panel pathogens. So they're all listed here on the right. But that's the current list, including mostly viruses, but also a couple of viroids. And at the bottom, you can see Xylella fastidiosa, and we do a universal phytoplasma test. And so this is a combination, as I mentioned, of high throughput sequencing, which should detect all known and also all novel viruses and virus variants. And then also our more targeted testing, which would be QRT-PCR or qPCR. And these tests all have higher sensitivity, higher specificity, and another benefit of them is that you can multiplex, or you can test for multiple pathogens at a time. So currently, the testing that we're doing through the Budwood program is ELISA testing. That's an antibody-based test that we can't multiplex, and it's not as sensitive or specific as these more advanced techniques. So one experiment that we are going to be doing this spring with the Budwood program is we're going to test a subset of those samples by qPCR to see if we're detecting earlier infections by qPCR that we're missing using ELISA. And if that is the case, then we'll begin the process of switching over our diagnostics for the Budwood program from the ELISA testing to the qPCR testing. And so, as I mentioned, if we do find a virus in these accessions, then there are elimination therapies that can be conducted on the infected selections, and usually this is a combination of heat therapy and then also followed up with meristem tip tissue culture. So we actually just before the Christmas break, we went out to the Clean Plant Center Northwest and we got trained on some of these techniques. We're working on developing the infrastructure for this for heat therapy and for tissue culture in my lab. So we just got a new heat therapy chamber, also right before Christmas. It was a great Christmas gift. And so once we get those capabilities going in the lab, then we'll be able to do that on any new accessions coming out of breeding programs or any public selections that are available in the U.S. And the reason why we want to maintain this in the Southeast, I mean, the other clean plant centers have been doing this for decades. The ones at Washington State and FPS, but those are both West Coast. So there's no one right now doing this on the East Coast, and a lot of the plants that get sent to, for example, Washington State to keep them in foundation, they tend to not make it. They don't like the cold up there. So we really need this in the Southeast, where a lot of these cultivars are meant to be. So again, once it goes through virus elimination therapy and we know that we have the clean plant, then we can maintain it in foundation. We do have an active foundation already, started by Dr. Simon Scott, and this is maintained at the Musser Farm at Clemson. So these are virus-negative, clean prunus cultivars that are important to the Southeast, and we have them maintained in both indoor and outdoor foundations, again, under these conditions that kind of minimize the risk of reinfection. They are also tested annually for a subset of those prunus panel pathogens. So we don't test for all 26 again, but we do test for the ones that we know are a risk in our area. And so this foundation collection will serve as a source of clean nuclear stock material that's available for propagation. So the indoor foundation right now, we have it set up so that the trees are in pots, and they're tested each year for Prunus necrotic ring spot virus, prune dwarf, plum pox, Xylella fastidiosa, peach latent mosaic viroid, the universal phytoplasmas, and then also we're testing a subset of these by high throughput sequencing each year. So these are tested very rigorously annually. And that's because it takes a lot of work and a lot of time to create a clean plant. So once it's clean, we want to keep it that way. And then the outdoor block, we have mostly as a way to increase the number of buds that can be collected from these trees. So this is kind of I like to call it a G1.5, not quite G2, but because it's maintained at the same foundation standards. But there's a big push in the NCPN right now to move all the foundation material indoors. But we just can't get the number of buds that would be needed by the industry from those the couple of trees that are maintained in the indoor block. So we're establishing this outdoor block as well. So these were planted last year, and we test these for a lot of the same viruses. We have to test for, the tomato ring spot and tobacco ring spot viruses. These are Nepo viruses, again, that are vectored by the Dagger nematodes. And so we want to test for them because they're in the soil outside. And then also peach light and mosaic viroid, nectarine virus M, and then Xylella and the phytoplasmas. And this is our current list of what we have in the Prunus Foundation right now. So I listed the peach cultivars that we have in the screenhouse and the field block. We maintain 2 to 3 trees in the screenhouse, and then anywhere from 8 to 12 trees in the outdoor block. And this probably isn't going to provide the number of buds that are needed by the industry each year, but at least it's a start. And we can. It's kind of like a bit of an insurance policy too, that if something were to happen, like a plumpox outbreak or I don't know, something where material was needed in a hurry, then we have that source at the mustard farm. And so we have several rootstock cultivars and a couple of plums. We are also getting a couple of new ones. There's one from the NC State breeding program, from Denny Werner's program. It's called Montgomery Gold. We'll be getting that one this year, and then also MP29. We'll be getting that rootstock this year too. Excuse me. So I just want to give a bit of a side-by-side of the Budwood program and the foundation, because these are complementary programs, but there's some big differences. So in the Budwood program, this one has 4 participating nurseries and several participating growers each year, but it's essentially kind of limited to that group. The foundation is a public program. It's funded by the NCPN. Anybody can request material from the foundation. In the Budwood program, the trees are tested for only 3 viruses, but in the foundation, they're tested for that whole panel. And then in the Budwood program, though, we can get a lot more buds off those trees. So because we have these larger blocks of 50 to 100 trees, and they're full-size production trees, they're able to provide the maximum amount of budwood compared to the foundation plants, which we just can't get enough buds off of those. And then with the Budwood program, it's a lot quicker to establish a block. So essentially, we just pick it, the growers show us where the trees are, we mark them, and we can test them that spring. Again, only for those 3 viruses. And then with making a foundation plant, it can take several years, especially if the plant is infected and needs to go through virus elimination therapies. So I just want to mention real quickly this experiment that we have going on to determine if Xylella fastidiosa is transmissible through grafting in peach. Again, this is a collaboration with Phil Brandon and his student Kendall. So we completed tea budding using Xylella fastidiosa infected material in the summer, and we'll do chip budding here in a couple of weeks to see if there's a difference in transmissibility between those 2 types of budding. And I just want to conclude by highlighting a recent article that published in Plant Disease, where the focus is on the economic value of these clean plant centers, essentially. So they consistently show returns on public investments ranging from 10 to 1 to 150 to 1. So these clean plant centers will hopefully be able to continue doing this work, and we'll continue to work with our peer centers in Washington and California. I just want to say a quick thank you to my lab, to our sister centers at Washington State and UC Davis, especially Scott Harper and Maher Al-Rawani, the Clemson Peach team, Mustard Farm crew, the Peach Councils, and of course the NCPN. We have a fancy new website, so check it out, cleanplant.clemson.edu. And that's all I have. Thank you.